So we're talking about the physiology of muscle contraction. And there has to be a mechanism by which we can regulate when a muscle is going to contract and when it doesn't. So we talked about how muscles can be stimulated voluntarily by sending an impulse via a motor neuron. And motor neurons release into the synapse with skeletal muscle fibers acetylcholine. So that's the excitatory neurotransmitter that will then cause the muscle to contract. So let's talk about why we have to have this signal in order for the muscle to contract. So we need to talk about some regulatory proteins. So the regulatory proteins that we're going to talk about are tropomyosin and troponin. Tropo means upon. So this tropomyosin is a protein that covers the myosin binding sites on the actin. So if tropomyosin is in place, then myosin cannot bind to the actin and muscle contraction cannot take place. So tropomyosin is a regulatory protein within our muscle fibrils. So within our myofibrils, the organelles within the muscle fiber. So troponin is actually attached to tropomyosin. Tropomyosin. And it is going to bind calcium ions. We have not talked about calcium ions. We did talk about calcium when we were talking about bones, and that there's calcium in our blood. And one of the reasons why there's calcium in our blood is that both our nervous system and our muscular system use calcium ions. So calcium is super important for the function of the muscular and the nervous system, but also obviously for the strength of our bones. Okay, so let's look at some diagrams, shall we? And when we look at the myofibrils, remember that these are the organelles, we see that this motor neuron is innervating my muscle fiber, and it is going to release acetylcholine. Acetylcholine is then binding to these ion channels. When they bind to the ion channels, it causes sodium ions to flood in to the inside. And that is actually going to cause depolarization of your muscle cell, of your muscle fiber. Okay, so let's look at it in more detail. So this is like a blown up picture of what is happening inside of your myofibrils. This is your motor neuron. It is releasing acetylcholine. It binds to those sodium ion channels. And then what happens is that there is a depolarization, and this depolarization causes calcium to be released into the inside of the myofibril. So calcium is stored, but if it depolarizes, then calcium is released. These calcium ions then interact with those regulatory proteins. So if calcium is not present, the muscle is relaxed. This right here is your tropomyosin. Notice that it is covering these binding sites on the thin filament. This is troponin. This is what is going to bind the calcium ions. So the calcium ions are released here, and they bind to troponin, and the binding causes a change in the shape of the protein. Can you believe all of this crazy biochemistry is happening inside your cells every time you contract a muscle? And how did they even figure this out, right? So just be amazed. Okay, so we have these tropomyosin, and notice that it moves, and now I have these binding sites that the myosin heads can bind to. The myosin heads bind, and so if calcium is present, it will cause the sarcomere to shorten and the muscle contracts. So how can I summarize this in a few words as possible? So let's look at our next page. Okay. So I'm going to make a list. 1. Okay. Depolarization of the muscle fiber causes the release of calcium ions. And they have it like 2+ instead of plus calcium ions into the space, okay, into the like where the myofilaments are. Okay. Calcium ions bind to troponin, causing tropomyosin to move. This opens up those binding sites. Okay. This allows, maybe I should put that as 3. This allows 3. This allows the myosin heads to bind to actin and the sarcomere shortens. Okay, I think I got everything in there. So let's go back and look at this image again. Okay, this is the structure in which the, excuse me, I need to point to it. This is the structure where the calcium ions are stored. You do not need to know the name of that. That is actually called the sarcoplasmic reticulum, but don't worry about knowing the name of that. So when depolarization occurs, these calcium ions are released and then they interact with the regulatory proteins. So they actually bind to troponin, which is the smaller molecule. The tropomyosin moves and the binding sites open up. And so these myosin heads bind here. And then the energy that is stored in those myosin heads, which we'll talk about next, causes a pivoting of them. So they pivot and the whole thing slides together. So the sarcomere shortens by the filaments sliding in relationship to one another. And so this is a sarcomere right up here that is not contracted. It's not shortened. This is the sarcomere that is shortened. And you'll notice that it is like, there's lots of overlap between the actin and the myosin, and the muscle can't shorten anymore. So it's at the most force that it could possibly generate. It's contracted. Okay. So the next thing is talking about how is all of this powered? Where do we get the energy to cause these filaments to slide? And so that is the next lecture.